Welcome to the DX Today podcast, your daily deep dive into the AI ecosystem. I'm Chris, and joining me as always is Laura.
Thanks, Chris. Today I want to talk about the one corner of the AI world where the marketing promises and the measured outcomes have drifted the furthest apart, and where the data has finally gotten good enough that we can say something honest about it.
That sounds like a setup for bad news, so let me brace myself and just ask you directly which corner of the ecosystem you're aiming at before you start dismantling it in front of everybody.
Drug discovery, specifically AI-originated therapeutics. And the headline statistic that all of biotech is chewing on right now is this. More than 173 AI-originated drug programs are currently sitting in clinical trials, and exactly 0 of them have been approved.
Hold on, because I want to push back before we go any further. 0 approvals sounds devastating, but drug development famously takes 10 to 15 years, and this entire field is barely a decade old. Is 0 actually damning, or is it just early?
That's the correct objection, and I want to give it full credit because the timeline defense is legitimate. Most of these programs entered the clinic within the last 3 to 5 years, and nobody serious should expect approvals yet from a cohort that young. The real story is one layer down in the phase-by-phase breakdown.
Then walk me through the phases, because I think a lot of our listeners know roughly what a clinical trial is, but not what each individual stage is actually testing for or why the distinction matters so much here.
Phase 1 is primarily about safety and tolerability in a small group of humans. You're asking whether the molecule behaves predictably in the body, whether it gets absorbed properly, whether the liver handles it sensibly, and whether it does anything obviously toxic at reasonable doses.
And how are the AI-originated candidates performing on that particular measure? Because I have a strong feeling this is the part of the story where the technology comes out looking genuinely impressive.
They're doing extraordinarily well. AI-discovered molecules are hitting phase 1 success rates somewhere in the range of 80 to 90% against a historical industry baseline that sits closer to 52 or 66%, depending on whose dataset you happen to trust.
That is an enormous gap. And if I were writing the press release, I would stop right there and go home. But explain to me why a machine learning system would be so specifically good at that one hurdle.
Because phase 1 is at its core a chemistry problem wearing a biology costume. You're optimizing molecular properties, solubility, metabolic stability, how the liver processes it. Whether it crosses membranes, whether it quietly accumulates somewhere, it absolutely should not be accumulating.
And those are all things you can learn from data in principle, because the industry has decades of molecules with carefully measured properties sitting in databases, just waiting for a model to come along and find the pattern.
Exactly right. That's a supervised learning problem with abundant labeled data and reasonably stable underlying physics. Generative chemistry models can propose structures satisfying a dozen competing property constraints at once, which is something human medicinal chemists find genuinely grueling and slow.
So the machines are better at the chemistry than we are, or at least dramatically faster at searching the space. Now tell me what happens to those same molecules when they walk into phase 2.
They fall off a cliff. Phase 2 success rates for AI-originated candidates land at roughly 40%, which is statistically indistinguishable from the historical average for conventionally discovered drugs. Every bit of the advantage simply evaporates at that boundary.
Say that again, because I want the audience to catch the full weight of it. The advantage doesn't shrink, it doesn't get cut in half, it just vanishes entirely. And what is phase 2 testing that phase 1 wasn't?
It vanishes entirely, and that single transition is the most informative number in the whole field. Phase 2 tests efficacy. Does the drug actually make sick people better? And that question doesn't care how elegantly your molecule was designed. It depends entirely on whether the target you aimed at genuinely matters for the disease.
So the model can build you a beautiful key, but it can't tell you whether you picked the right lock. And picking the right lock is the part that determines whether anybody actually gets healthier.
That is a perfect way to frame it, and I'm going to steal it permanently. Target validation is the bottleneck, and target validation is fundamentally a biology question about causal mechanisms inside living systems that we still do not understand very well.
How much of phase 2 failure traces back to that specific problem as opposed to safety issues, or manufacturing, or trial design, or any of the other dozen ways a clinical study can go sideways?
Roughly 70% of phase 2 failures come down to lack of clinical efficacy. Not toxicity, not manufacturing, not enrollment problems. The drug simply doesn't work well enough. And that almost always means the underlying biological hypothesis was wrong from the beginning.
All right, that's the pessimistic frame, and it's well supported. But I know there are specific programs that people point to as genuine proof this works. So let's give the optimists their day in court.
The strongest case by a wide margin is rentosertib from Insilico medicine. It's a TNIK inhibitor for idiopathic pulmonary fibrosis, which is a brutal progressive lung scarring disease with very few good treatment options available to patients today.
And what makes that particular molecule the flagship of the entire field, as opposed to any of the other 170-odd programs currently sitting somewhere in the clinical pipeline?
Because it's the first case where generative models did both halves of the job. The AI identified the novel biological target, and then the AI designed the chemical structure to hit that target. Both ends of the problem, from scratch, with no human handoff in the middle.
That distinction matters enormously, doesn't it? Most of what gets marketed to the public as AI drug discovery is really AI-assisted optimization of a target that some human biologists proposed a decade earlier.
Correct, and that conflation is responsible for an enormous amount of the hype inflation in this sector. Rentosertib is the rare case where the strong claim happens to be the accurate claim, which is precisely why everybody in the industry watches it so closely.
So give me the numbers. What did the preclinical program cost, and how long did it take from initial target hypothesis to a nominated candidate? And I'll register my skepticism about company-reported figures in advance.
Insilico reports roughly 18 months and approximately $2.6 million, from target hypothesis to preclinical candidate nomination. Traditional programs routinely run 4 to 6 years at that stage. But your skepticism is warranted, because Insilico spent hundreds of millions building the platform that produced those 18 months.
So the marginal cost per program is real, but the amortized cost is a completely different number. Now, the part that actually matters to patients. What did the clinical results look like, and were they published somewhere serious?
Nature Medicine published the Phase 2 results in June of 2025. In the 60 milligram once daily arm, patients showed a mean improvement of plus 98.4 milliliters in forced vital capacity, which is the standard lung function measure for this disease.
I need you to translate that for me, because I understand it's a lung capacity number, but I have absolutely no intuition for whether 98 milliliters is a rounding error or a genuinely meaningful clinical result.
It's meaningful, and the comparison is what makes it striking. The placebo group declined by an average of 20.3 milliliters over the same window, which is exactly what you'd expect to see in a relentlessly progressive fibrotic disease.
So the untreated patients got worse, the way this disease always makes people get worse. And the treated patients actually gained lung capacity rather than merely slowing down the rate at which they were losing it.
That's the signal. An improvement rather than mere deceleration is genuinely unusual in this indication. The existing approved therapies for pulmonary fibrosis mostly slow the rate of decline. They do not typically give anybody back the capacity they've already lost.
Now give me the caveats, because a phase 2 trial is small by design, and I don't want us accidentally recording a commercial for a company that neither of us works for.
Absolutely necessary caveats. It was a small early-stage study conducted primarily in China, carrying all the usual limitations of dose-ranging trials that were never statistically powered to prove definitive efficacy in the first place.
And where does the program stand today? Because I gather this one has moved considerably further along than anything else currently carrying the AI-discovered label.
It has advanced toward Phase 3, which makes it the most clinically mature AI-discovered candidate anywhere in the world. And Phase 3 is the honest test, because that is exactly where optimistic early signals historically go to die.
Let's widen the lens a little. Is there anybody else with a credible clinical readout? Or is this entire field currently resting its whole reputation on one single molecule from one single company?
Recursion has a real one. Their compound Rec 4881 showed a 43% median reduction in polyp burden over 3 months in a Phase 1b/2 trial for familial adenomatous polyposis, which is an inherited cancer predisposition syndrome.
And I understand Recursion's entire scientific approach is philosophically different from Insilico's, which seems worth unpacking because these aren't just two companies racing to do the exact same thing.
Very different. Insilico leans on generative chemistry and knowledge graphs to reason about targets. Recursion built enormous automated microscopy labs and learns from images of cells, essentially doing pattern recognition on biology itself rather than on molecules. And honestly, neither approach has separated from the pack yet.
Meanwhile, the money keeps arriving regardless of that blank scoreboard, which tells me the large Pharmaceutical companies are making a bet that doesn't actually depend on any of these specific programs succeeding.
They are, and the newer deals are structurally different. Sanofi announced a multi-year partnership with Okin specifically to develop agentic AI systems for pharmaceutical research, which is a meaningfully different ambition than simply generating better molecules faster.
Unpack agentic in this specific context for me, because that word has been stretched so far over the last 2 years that I honestly no longer know what anybody means when they say it.
Here it means systems that plan and execute multi-step research workflows instead of answering one isolated question. Propose a hypothesis, design the experiment, interpret the result, revise the hypothesis, and loop without a human sitting in every single cycle of that process.
Which is aimed squarely at the bottleneck you identified earlier. If target validation is the real problem, then automating the hypothesis testing loop is arguably the only thing that could plausibly fix it at scale.
That's the strategic logic, and I think it's sound, even though the execution is completely unproven. You cannot chemistry your way out of a biology problem. You have to actually run more experiments faster, with better feedback between them.
Let's turn to the regulators, because none of this reaches a single patient without them. What is the FDA actually doing about artificial intelligence in the drug pipeline right now?
The agency's draft AI guidance is expected to be finalized this year, and the core requirement is that sponsors build credibility assessment plans for high-risk applications. That means documenting model architecture, training data, and governance, and proving the model is fit for one specific context of use.
The context of use framing seems like the important part, because a model that predicts toxicity beautifully for one chemical class might be useless for another, and the agency clearly understands that. Is there a concrete example of them engaging with an actual tool?
Yes, and it's a good one. The FDA has accepted a letter of intent to evaluate an AI tool that predicts drug-induced liver injury, which is one of the top causes of both development failure and post-market withdrawal. It's the ideal beachhead, honestly.
Let me play devil's advocate one final time before we wrap. Is this whole field just another hype cycle waiting to deflate? Or is something real quietly accumulating underneath all of the noise?
Something real is accumulating, but it's far narrower than the marketing suggests. AI has made the design and optimization stage dramatically faster and cheaper, and that genuinely is not nothing. It's just not the stage where drugs actually fail.
So what would actually change your assessment? Give me a falsifiable test, because that's far more useful to our listeners than any general optimism or general pessimism the two of us could offer them.
Here's mine. If AI-originated candidates start beating that 40% phase 2 baseline in a sustained way, across multiple companies and multiple disease areas, then the technology has genuinely reached target biology and every assumption in this conversation changes.
And until that happens, we're essentially watching a very expensive, very fast front end bolted onto a back end that still fails at precisely the same rate it always did. What are you watching next?
The rentosertib phase 3 readout, without question. One approval would reset every conversation about this technology overnight, and one high-profile failure would reset it just as violently in the opposite direction. That's the whole field in one trial.
That's a clean place to land, and I appreciate you keeping this grounded in the actual numbers rather than the press releases, which is much harder than it sounds in this particular sector.
My pleasure entirely, and I'd add that the honest version of this story is more interesting than the hype version anyway, because the gap between chemistry and biology tells you something real about the limits of intelligence itself.
That's all for today's episode of the DX Today podcast. Thanks for listening, and we'll see you next time.